All right, so how do we know that this special repolarization pattern we see in these cardiac contractile muscles is important to its function and what kind of controls it? So in a review of studies done by Saw and colleagues, we see that the regulation of cardiac excitation and contraction coupling is modified and controlled by the transient outward potassium current. Okay, so that potassium that we see go out initially in that first phase of repolarization is what starts this process to make it effective. And so we have here our potassium that's moving out of our cell. We have our calcium that's moving into our cell. And then they also propose these calcium-sodium exchanger sites on the cell that are going to allow more intracellular calcium, as well as, in order to accomplish that excitation-contraction coupling, we have the calcium that is going to be released from our sarcoplasmic reticulum per general muscle contraction. So as they went through this study, they looked at several factors that could affect this contraction coupling process. And what they found was that the fast phase 1, so that initial phase of repolarization, right here, almost making this little notch here, was really important to controlling the rest of the plateau, the rest of that repolarization phase, and ultimately allowing enough calcium to be released so that that excitation contraction coupling could happen properly during the repolarization phase of the action potential. So what you see here is this is your fast phase that is normal, okay? And then you have this slow phase, which is a modified phase. What happens in that can be done artificially in model organisms by controlling gene expression, or in diseased tissue in humans. And what we see is that the L-type calcium channels that are triggered and allow for calcium to be released in the cell to allow for that excitation contraction coupling to be activated and function properly, we see this nice quick trigger of those calcium channels to release calcium, whereas when we do not have that initial phase, we don't get that quick release of calcium as we should. At the very bottom of this figure, we see surface plots that show recruitment and temporal synchronization of calcium release in those myocytes. And what we see is when we have that fast phase 1 repolarization, we have ample recruitment and coordination of that calcium release, whereas if we do not have that fast first phase, then we see a loss of recruitment and then less synchronization of calcium release to start that excitation contraction coupling. So ultimately, what this study showed was that a slowed repolarization without that initial fast phase repolarization mirrors the calcium release that we see in diseased tissue in human cardiac myocytes, but has been displayed in these model organisms